Hello, my name is Ben, and my video project is about a protein called FOXP2. For a bit of a background, FOXP2 is encoded by a gene of the same name, and it is a transcription factor for other genes. FOXP2 plays a role in speech and language development, as well as in brain and lung development. This protein was first discovered when the KE family from Britain were observed to have a high frequency of the same speech disorder. It was discovered when affected and unaffected members of the KE family and an unrelated affected individual were compared. The location of the gene was first discovered from that unrelated individual on chromosome 7 and was later identified as FOXP2. Structurally, FOXP2 can be monomeric or dimeric. All proteins of the FOX family, including FOXP2, have a motif called the winged helix, which features a bundle of 3 or 4 alpha helices capped by 3 beta sheets. FOXP2 is comprised of 4 alpha helices capped by 3 beta sheets. Helix 3 plays an important role by being the mediator for DNA recognition. Helix 3 strongly binds to DNA and wedges into the major groove. From the online databank, this model includes the DNA, the monomeric FOXP2, and this unrelated NFAT protein, which is also a transcription factor. The different colors represent different secondary structures, and this will be explained in the next few slides. The protein up here is our protein of interest, FOXP2, and the proteins here are called nuclear factor of activated T cells, or NFAT, as mentioned earlier. Our focus will obviously be on FOXP2. Like I mentioned before, FOXP2 has 4 alpha helices and 1 3-10 helix and 3 beta sheets. The secondary structures are color-coded. The yellow represents beta sheets and the purple represents alpha helices. You may have noticed that the helix 4 has a different shade of purple, which is right here. Helix 4 is found in the turn between helix 2 and 3 and is actually the 3-10 helix mentioned earlier. And here's a closer look at helix 3 wedged into the major groove of DNA. Monomeric FOXP2 binds to DNA strongly. The residues between helix 3 and DNA form strong hydrogen bonds and hydrophobic interactions, which are stronger than that of dimeric FOXP2. Dimeric FOXP2 is formed when 2 monomers form a domain-swapped dimer. When 2 monomers form a dimer, they exchange helix 3s and are then stabilized by complex aromatic interactions. However, the dimeric form has significantly fewer interactions with DNA. Here is a short GIF of the domain swap between 2 monomers. 2 beta strands of one monomer interact with helix 3 of the other monomer and forms one straight 15 amino acid sequence. And here is another look at a dimeric FOXP2 protein. Here's the locations of the 2 helix 3s of the dimer, and this right here is the long 15 amino acid sequence between helix 2 and 3. The dimeric form is stabilized by aromatic interactions between the 2 monomers. However, a monomer's binding to DNA is much stronger than the interactions between a dimer and DNA, which can be seen here in this comparison. The disease associated with a mutated FOXP2 gene is autosomal dominant. However, every affected individual were heterozygous for the mutant allele. Having one copy of the mutant allele caused reduced vocalization in mice. Having 2 copies caused death in mice due to the underdevelopment of their lungs. And here's the pedigree of the KE family that shows data consistent with a dominant allele. About half of the family is affected, and which is what you would expect from a cross between a heterozygote and a homozygous recessive. So the FOXP2 gene also provides some insight on human evolution. FOXP2 was sequenced in humans and FOXP2 from several primate relatives. Across different apes and monkey species, the FOXP2 sequences are identical. Interestingly, humans and Neanderthals both have identical mutations to FOXP2. A threonine at position 303 to asparagine, and an asparagine at position 325 to serine. Perhaps this gene is what sets apart human speech to animal noises. Here are my sources. Thank you for watching.